In today's video we shall discuss about isoelectric focusing. But before that, do subscribe to my channel and let me know your opinions and suggestions in the comment box. Isoelectric focusing. IEF is a technique used to separate and analyze proteins based on their isoelectric point, which is the pH at which a protein has no net charge. IEF is a type of electrophoresis, which involves the migration of charged molecules in an electric field. In IEF, a protein sample is applied to a gel containing a pH gradient, which is created by mixing different buffering agents. The principle of isoelectric focusing is based on the fact that charged molecules, such as proteins, will migrate in an electric field towards a pH where they have no net charge, that is their isoelectric point. In IEF, a protein sample is applied to a gel containing a pH gradient, which can be created using different buffering agents. The gel is typically a long, thin strip that is mounted vertically in an electrophoresis apparatus. When an electric field is applied across the gel, the proteins migrate towards the pH region that matches their isoelectric point. As the proteins migrate through the pH gradient, they become increasingly concentrated in a narrow band around their pI. The separated proteins can then be visualized using different staining techniques, such as Koumassi blue or silver staining. Here are some common types of isoelectric focusing: gel-based isoelectric focusing, free-flow isoelectric focusing, immobilized pH gradient isoelectric focusing, capillary isoelectric focusing, and carrier ampholyte-based isoelectric focusing. Before moving on to the steps involved in isoelectric focusing, let us first see what a carrier ampholyte is. Carrier ampholytes are a class of buffering agents that are used in isoelectric focusing to create a pH gradient for separating proteins based on their isoelectric point. Carrier ampholytes are amphoteric molecules that can act as both acids and bases, and they can migrate along the pH gradient as the electric field is applied. Some examples of carrier ampholytes include polyampholytes, servolytes, and pharmalite. Moving on to the steps in isoelectric focusing. The major steps in IEF are preparation of IEF gel, addition of ampholytes, generation of pH gradient, sample application, running of electrophoresis, and finally visualization. First, the IEF gel is prepared by adding polymerized acrylamide solution on a glass plate, followed by the addition of ampholytes. After that, the pH gradient is generated by applying potential difference, followed by sample application. When the proteins in the sample meet their isoelectric point, their movement stops. Preparation of IEF gel and addition of ampholytes. Carrier ampholytes and riboflavin are dissolved in acrylamide solution. This mixture is then poured on a glass plate with attached spacers on sides. Riboflavin here helps in the polymerization of the gel. Then these plates are illuminated by bright light, which causes photodecomposition of riboflavin present in the gel and further produce free radicals. Free radicals thus produced causes polymerization or solidification of acrylamide solution, making it a gel. Generation of pH gradient. IEF gel thus prepared is then placed in electrophoretic chamber. For making anode, a piece of filter paper known as wick is soaked in the solution of phosphoric acid and placed on one side of IEF gel. For making cathode, a piece of filter paper is soaked in the solution of NaOH and placed on other side of IEF gel. Sample application and running of electrophoresis. Apply potential difference by turning on the power. The ampholytes form a pH gradient. Power is turned off. Sample is applied by laying on the gel, small squares of filter paper soaked in the sample. Power is turned on. Sample, here protein electrophoresis off the paper into the IEF gel. Protein move according to their charge towards their respective electrodes. Proteins movement. Proteins below their isoelectric points, positively charged, will move towards cathode. As they move, they meet their isoelectric point by capturing charges from ampholytes present in the gel. Their movement stop and become Zwitter ion. Proteins above their isoelectric points, negatively charged, will move towards anode. As they move, they meet their isoelectric point by capturing charges from ampholytes present in the gel. Movement stops and they become Zwitter ion. Different proteins stop at different places as all have different charges and all need equal opposite charges to meet their isoelectric points. This causes the separation of protein molecules. Visualization. Staining is done by Kumasi Brilliant Blue Stain. But it cannot be done directly because ampholytes will stain too, giving a totally blue gel. Gel is first washed with fixing solution, which is 10% trichloroacetic acid. This precipitates protein in the gel and allow much smaller ampholytes to be washed out. After that, gel is stained with Kumasi Brilliant Blue and then destained. Excision of protein band. The protein band of interest is excised from the gel and further analyzed using techniques such as mass spectrometry or Western blotting. Applications of isoelectric focusing. Protein purification. IEF is used for the purification of proteins from complex mixtures. Proteins can be separated based on their pi and then collected for further analysis. Proteomics. IEF is widely used in proteomics to separate complex protein mixtures. It is often used as a first-dimension separation technique in two-dimensional gel electrophoresis along with SDS page. Disease diagnosis. IEF can be used to detect abnormal proteins in body fluids, such as blood or urine, which can indicate disease or disorders. For example, the presence of specific proteins in cerebrospinal fluid can be used to diagnose multiple sclerosis. Drug development. IEF is used to analyze and characterize protein-based drugs, such as monoclonal antibodies, to ensure their purity and quality. Biotechnology. IEF is used in the production of recombinant proteins, such as insulin, by separating and purifying the protein from a mixture of other proteins. Advantages of isoelectric focusing. High resolution. IEF offers high resolution and can separate proteins with similar molecular weights but different pI values, minimal sample loss. IEF is a non-destructive technique and does not involve any physical or chemical changes to the proteins, thus minimizing sample loss. Large dynamic range. IEF has a large dynamic range and can separate proteins over a broad range of pI values. High reproducibility. IEF is highly reproducible, making it a reliable technique for protein analysis and purification. Can analyze a wide variety of proteins. IEF can analyze a wide variety of proteins, from small peptides to large proteins and protein complexes. Disadvantages of isoelectric focusing. Time-consuming. IEF is a time-consuming technique that can take several hours to complete. Complexity. IEF is a complex technique that requires specialized equipment and skilled personnel. Sensitivity to impurities. IEF is sensitive to impurities, such as salts or detergents, which can interfere with the separation and affect the results. Limited sample loading capacity. IEF has a limited sample loading capacity, and large amounts of sample can result in overloading or distortion of the separation. Nonlinear response. The relationship between protein pI and migration distance is nonlinear, which can make it challenging to accurately determine the pI of a protein.